Thank you for the organizers to making possible this outstanding year of the Phage meeting, and especially also for inviting me and have the opportunity to talk today about modeling capsids. This is within the field, emerging interdisciplinary field of physical biology. And let me start the talk trying to justify a little bit why many physicists are becoming more interested, very interested in the structure and properties of viral capsids. So I think that the best way to do that is with an animation from the Rosman lab of a Phage T4 virus. Here you can see the Phage T4, it's rambling around maybe in your gut, trying to find an E. coli, and then the fibers find the surface, and then you have the base plate that triggers the Mertens-City transition that translocates the DNA, and at the same time you have these proteins that start to digest the membrane, and the infection begins. I mean, how cool is that? And from, well, I mean, most of the data here is structural, based on cryo-EM and X-ray, but the animation is a little bit artistic. But the idea from physics is that these high-resolution experiments and other type of techniques are showing that bacteriophages are sophisticated nanomachines, and the more we learn about it, we realize that they rely on relatively simple physical mechanisms to succeed during the life cycle, like from protecting the DNA during when they search for another host, and then injecting the DNA, the mechanisms that they use for the assembly of the particles or the egress of the particles, like we saw, for instance, at Ryan talk, using the Hauling critical nucleation transition to start the lysis. So in addition to these mechanisms, there is another interest in physics to study the structure of viruses, that is especially bacteriophages, that they can sustain, some of them, 50 to 100 atmospheres of internal pressure. It's like you can use them as nano containers. You can use them to have nano reactions inside. You can use them as nano templates to do other kind of materials. So in order to progress on this, on the understanding of these properties, we realize that it's very important to develop theoretical and computational models that can help test ideas and have a solid framework to keep progressing. So here I divided the talk in a couple of blocks. The first one, I'm going to just give a glimpse on structure and assembly methods for viral capsids, and then encouraged by Forrest, I will give you maybe a couple, we'll discuss a couple of challenges that should occupy people like me for the next millennium. So let's see, just many viruses, including bacteriophages, adopt spherical or quasi-spherical structures on their capsids, and they have icosahedral symmetry. And then in these capsids, you have, maybe I should use the pointer. These capsids, you have 12 pentamers that nucleate the proteins on the vertices of this icosahedron, and then you have regular hexagonal networks of hexamers that are in the 20 triangular facets of the icosahedron. And the mathematical properties of these capsids were derived originally by Casper and Kluck, and they found the index T helped to classify all these structures, and here you can have the lambda-like HK97 that Jack Johnson was talking yesterday is T equals 7 and has around 420 proteins on the capsid. So these structures are highly symmetric and has motivated developing a lot of models, from continuum models to coarse-grained models to high-resolution models, including all-atom simulations. And these models are trying to explore the details on the structures of the virus, the mechanical properties of these viruses. And one of the properties that have been found is that we observe that these icosahedral structures have associated very well-defined free energy minima. And when we analyze the energy interactions between these proteins. So this means that at least thermodynamically, this justifies why many viruses that are completely different can adopt these similar symmetries on different sides. One specific application, besides these icosahedral symmetry, structures also can adopt two types of shapes, let's say. Usually they can adopt a more spherical shape, sometimes they adopt a more polyhedral shape. And this is a property that bacteriophages like HK97 and other bacteriophages use during their maturation process when they're packaging their DNA to reinforce their capsid. So here, and this is one of the transitions that yesterday Jack Johnson was showing. In fact, this is data from Alasdair Steven, but he's been working a lot on this as well. And you can see that it goes from a more spherical to a more expanded polyhedral shape that is particularly reinforced to resist around 50 atmospheres of internal pressure. So there's been a strong effort using continuum theory to develop elastic models to explain the interactions between the proteins, which kind of interactions dominate this transition. Coarse-grained models have shown that there are specific T numbers that are more prone to favor this maturation. And high-resolution simulations have shown the fluctuations that are necessary to start triggering this transition. There's been a lot of effort on these spherical capsids, but as we saw in other talks, viruses can adopt all kinds of shapes. Archaea viruses in particular are amazing, but our efforts are focusing first on quite abundant capsids in phages as well, like 529 and T4 that are elongated structures, but we're just trying to develop the models to understand why these structures emerge, why phages adopt these structures, and which are the structural properties that this type of capsids give to the phage. So let's give also a brief, revise a little bit the concepts that we are studying in the assembly of viruses. One interesting thing about viral capsids is that many of them can be self-assembled spontaneously once you have the solution of proteins in vitro. And this has helped to study this self-assembly in vitro and found several common properties that you can observe in different kinds of viruses. And one of them is when you start the experiments, you have a lag time period where you don't observe production of capsids. Then, if you try to look for the intermediates that make the capsid, they're very difficult to capture. If you change the initial conditions of proteins, these dramatically change the production of capsids. So it's a strong dependence on that, and you need a minimum concentration to start to form these capsids. And when you have the fully formed capsid and you want to study how it disassembles, the conditions that you need to produce that are completely different from the ones that you would expect from the assembly. So to explain this, from a physics point of view, these properties fall on what we will call a first-order transition, where usually you have an energy barrier that separates two stages. One phase is the dissociated proteins, the other is the fully formed capsid. And this energy barrier helps to explain all these four properties that are observed on the experiments. So the barrier is the responsible for precluding the formation of capsids at the beginning, having the lag time. If you have intermediates that are on the top of the barrier, if they surmount this barrier, then they follow a downhill process, forming quickly a capsid. Otherwise, they dissociate again. And that explains why you barely observe any intermediates in the experiments. This barrier is a trade-off between the interactions of the proteins in the capsid and the entropy of the proteins that are dissociated. And this depends strongly on the concentration of proteins. So if you increase this concentration, this barrier is lower, and that favors the formation of capsids also as it's observed. And then here, when you have favorable conditions of assembly, the barrier to jump from the complete capsid to dissociate is much higher than when you form the capsid, and that explains the hysteresis that you observe when you compare the conditions of assembly and disassembly. This first order transition, it's analogous to what we observe when you form a drop, a water drop. And this is a well-studied process in physics, and it's studied using classical nucleation theory, for instance. And there's been an effort in the field to develop classical nucleation theory for viral capsids and to mathematically formalize this assembly process and compare to experiments. And at the same time, there's been an effort developing simulations that for computational reasons need to use simplified models, coarse-grained models, but they've been able to reproduce, to study different type of capsids, and this has helped to clarify some of the hypotheses that we use in the classical nucleation theory in a more visual way. However, we are still far from simulate like high-resolution structures, and some of the theories that we use for capsids don't capture yet the elements that we observe in bacteriophages, because in bacteriophages, instead of having just the proteins of the capsid assembled, you usually have, or in many cases, you have scaffolding proteins. So we still don't have a theory for having scaffolding proteins assembling at the same time of these proteins. We don't have those simulations either. So this brings me to the challenges that we were mentioning before. So I will give first a challenge that is more specific, and it's probably one of the lines that I would like to work on in my lab. And the idea would be, can you predict the structure of all phages when you have when you sample any kind of environment? Like for instance, you get a sample from human microbiome. Let's assume that metagenomics, proteomics, and protein folding are advanced enough that you can identify perfectly all the phage genomes, and you can provide the 3D models of the structural proteins. And that's where I start. So I would say that it's a long challenge, because this is probably the present and the near future is this, I guess. And we're trying to understand this from a different direction, and the idea is now you assume, okay, I have these structural proteins, and this is the two biggest challenges, I think. Is, okay, we have a bunch of structural proteins, but these proteins need to assemble in subparts of the viral capsid. You have to form the capsid, and this has hexamers, this has pentamers, you have accessory proteins. These have to form base plates, you have a portal, you have a connector, you have the tails, you have fiber capsids, fiber tails that you don't see, that should be here. These are many possible combinations that when you have these proteins, if you don't know exactly where they go, this is already very challenging. But on top of that, you need to combine these things to make the phage, and you need to understand how this assembly needs to process in order to have the right structure. Simulations nowadays that sometimes deal with small parts of here, or the most sophisticated simulations, we get barely nanoseconds. And it's very difficult. I mean, and we cannot simulate processes that take long in high-resolution models. So we are really far on that. We need to refine the models. We need to be more efficient computationally. And we need to learn more in general, because when you want to reproduce the assembly, you probably need to explore different ionic conditions, different pH conditions, different concentrations of proteins, so you can get the right structures. Of course, if you end up having this tool, you can ask hypotheses about, okay, what's happening on the rapid evolution that you have in this genome. Then, of course, if you want to evaluate what is happening on that evolution structurally, and you have to do experimentally that, you're not going to be able to to answer those questions quickly. So this is why I think that theory and simulations need really to put an effort on that. And then you could probably evaluate what is happening on the changes on the structures. You can test hypotheses, or you can use this pipeline, this part of the pipeline, to, instead of analyzing a sample, you can just ask the questions of, if I want to develop a specific nanotechnological biomedical applications, which are the properties of phages that I need to achieve? Okay, so let's just try to generate that, at least on an in silico way, and at least we have good candidates. Following this pipeline and motivating that modeling needs to all to jump on an ecological level, on a realistic way, this is, let's say, a more open challenge that I suggest is, okay, we are pretty aware that Humans is having a huge impact on the planet. Our population is still growing almost exponentially. Our cities are growing, not only on extension, also vertically. Especially in the last century, we have increased the production of modern materials, concrete, metals, electronic devices. We have increased the emission of gases, CO2, methane. So what happens in the planet when there is a change like this one is what had happened in the past, at least, is that bacteria evolve and they just try to develop the ecosystem accordingly. And that means that at some point, sooner or later, we're going to have like probably some sort of new microbiomes growing everywhere. And if you think about it, that can be like a threat. Like imagine that bacteria get so they develop new pathways as they did, for instance, when they develop photosynthesis that changed the atmosphere, that changed everything. So you would think, okay, you're a threat because these bacteria could be degrading the buildings, they could degrading the computers, the bridges, everything. So my research challenge would be, we need to get ahead of that. We cannot wait until this happens and then it's bad. And we need to develop theoretical and computational models that are able to predict when these things could happen, when these things, which direction they could take, which kind of phages should we develop in order to regulate these changes on the ecosystem, so at least we keep an atmosphere that we can breathe. And maybe if the planet becomes like completely that we cannot stay here anymore, so at least we can also develop an atmosphere on the space adventure that we will have to take. And I believe that phages, it's our tool for survival in general, because it's what we can use to communicate with bacteria, and bacteria is essentially what dominates the planet. So thank you very much. I will just leave the people that has taught me so far and the people that I will be working here in SDSU, and if you have any questions, I would like to answer you.